Hello, welcome back to Legal Research Institute. Don't forget to like, comment, share, and subscribe to my channel. Thank you all. So today we are at the site in which we want to collect our sample, and I want to do an enlightenment about our project topics. Our project topics, we are going to do antimicrobial evaluation and phytochemical screening of Carrisa spinarum, whether it has an antibiotics effect against some selected bacteria. So the selected bacteria will be like, we are going to use Klebsiella, we are going to use Staphylococcus, Strep, and E. coli. So now we are at the site. This is the tree. Its name is called spinarum in botanical name, but in English we call it conkerberry or bush plums. And according to the owners, like in Bra, it's been called drum. So it's called drum. And as we can see, we have different size of visions of the plants. As the first one, can you see? Okay, show the camera. You can see this one is half ripened. It shows red and green. It's half ripened. You can see this one is fully green. It shows that it's unripened one. It's just starting to ripe. And they'll come and see the last part. You can see it. It fell down. So it turns to like purple, dark red color. And this is the ripe one that I collected. This fruit is edible. It's very edible and like it's sweet in taste. But if you taste this one is half ripened. It's going to be sour, and you can see some chemicals coming out from it. So now we want to get it. And we are doing a group project. We are for in number. This is my other colleagues, so we are working on it. He's going to use the knife to cut it and put it inside the nylon. And another class like is a shrubs. Like what we mean by shrubs is like a small bush trees. They are very small in size and they have a fern. Look at their fern. Like you see, it can easily infect someone. Look at the fern. So the tree has a thorn, and yeah, it looks beautiful. Yeah, so this is how we are getting the sample. We are going to cut the tree so that we can get the fruits and also you see this one is an unripened one.
Hold this one first.
Okay. This one is a ripe one, this one is half ripened, and the one at my hands is ripened already. So that's how we're going to get the sample, put it inside a nylon. Then after that we take it to the laboratory for phytochemical analysis before we do the antimicrobial evaluation. Actually, you can cut like that.
I can.
Yeah. You have to be careful with the phone.
No, so
I think that's okay. That's quite enough. After we can buy. Okay. Now this is the continuation of the work. We are now in the laboratory. We want to wash the leaf and also the fruit to remove the dirt and also the dust around it. It's so different from the fruits, no? You should wash it together, no? And let's just add everything inside. So now after we wash the fruits and also the leaves, now we are going to dry it inside the incubators. So yes. Now we you don't need to. I'm gonna go to put this in the lab. Yeah, put it inside the oven. What's in the dust? So after we wash it, the leaf and also the plants, we have already dried one of the sample before inside the incubator. Let me to close it. Okay, as we can see, this is the same Carrisa spinarum leaf, and this is the fruit. So we want to dry it inside the incubator at room temperature so that we can proceed with our phytochemical analysis. So this one, after one, we are going to, these are the dried leaf. We are going to put inside the mortar and pestle so that we can grind it. Then we put the leaf that was dried inside the incubator, inside the motor and pistils, so that we have to grind it until it turns into into a powder. So after that, we are going to sieve it so that we get the powdered one, very light one, so that we grind and we pound those ones that are still heavy. After that, then we are going to weigh the powdered Carrisa spinarum leaf. We have already weighed the foil paper, which measures 0.75. So now we are putting the powdered leaf on top of the weight balance so that we know the actual weight of it. And it measures a total of 66.5. So after that, we also dried our leaf. We dry our fruit, and the fruit is still kind of watery, like it still adds water. So we are just going to put it back inside the incubator after we already grounded it. So as we can see, we are just spraying it on top of the foil paper so that we can put it back inside the incubator. Good day to you all. This is the continuation of our research work. We are done with pounding the Carrisa spinarum leaf, and for the fruit is yet to be dry, so we do it next. Next thing to do right now is for us to weigh it. We have already weighed it, and it shows like 67.3 grams. That is the total weight of the leaf. So after we have already weighed it, the next thing we have to do is to prepare our extract. And to get our extracts, we are going to work on two extracts. We are going to work on methanolic extract and what an aqueous extract. But not because this methanolic extract and aqueous extract, they are just a soluble solvent, a polar solvent, in which we use in the extraction of what of the chemicals inside the plants. So it means that we have to prepare a solvent in order for us to do the phytochemical analysis of what of any plants. As I've already said earlier, we're working on what on two extract. We're going to work on what on methanolic extract and aqueous extract. For our methanolic extract, to prepare our methanolic extract, we have to put it into a lot of consideration. The first thing is like we have to get a certain grams of what of the powdered Carrisa spinarum leaf. Then we are going to macerate it inside a what a methanol. If we are using 100 grams of this, we are going to macerate it inside a what a 250 mils of what of methanols. So, and the levels of what of the methanols and leaf depends on what on either the temperature, because as the temperature increases, rises, it allows the methanols to enter the plant cells and their membranes so that they can lead to the extraction of the chemicals inside. But if the temperature is too much, it can also lead to what the cell degeneration. So we have to be what very optimistic about the temperature after we macerate the powdered Carrisa spinarum inside the methanol. And in addition, the times, the duration we are going to use also has an effect in what in the extraction of the chemicals. But as I've said, we are only working on what on aqueous and methanolic extracts. So, and these two extracts, they are what the reason why we use them is because they are polar solvent. And what they mean by a polar solvents are those compounds in which they are partially charged positive and negative. So, and most of the word of the chemicals inside the plant, they contains the polar solvent. So it's only polar solvent that can dissolve a polar solvent. It means that we are using the methanols in order to be able to do the phytochemical analysis of the plant and extract it. And for the aqueous also, aqueous water is also what is also a polar solvent. We'll call them dipolar molecules because they have partially charged positive charge and partially charged negative charge. So water, acetones, buffer, and also the methanol can use for the extraction. So after we get our methanols, we macerate it inside this one, and I've already tell us what the factors that is going to determine the amount of methanol to be used is the temperature, the amount, the grams of the what of the Carrisa spinarum powder used, and also the temperature and also the durations. And as we have already known, most of the secondary metabolites of plants are what are polar. That's why we use the polar solvent in order to dissolve all these compounds and do their phytochemical extraction. Got it.